Good morning and welcome to all. Today we are going to discuss about Krebs cycle, and it is also known as TCA cycle or tricarboxylic acid cycle. So it is a series of chemical reactions used by all aerobic organisms to release their stored energy through the oxidation of acetyl-CoA. And this acetyl-CoA can be derived from carbohydrates, fats and proteins. In addition, the cycle provides precursors for certain amino acids as well as the reducing agent NADH, nicotinamide adenine dinucleotide hydrogens, that are used in numerous other reactions. And its central importance to many biochemical pathways suggests that it was one of the earliest components of metabolism. So it was one of the earliest components of metabolism and may have originated abiogenically. Even though it is branched as a cycle, it is not necessary for metabolites to follow only one specific route. At least 3 segments of citric acid have been recognized. The name of this metabolic pathway was derived from citric acid. It is a type of tricarboxylic acid, often called as citrate, as the ionized form predominates at biological pH that is consumed and then regenerated by this sequence of reaction to complete the cycle. So the ionized form of the citrate that is consumed and then regenerated by this sequence of reaction to complete the cycle. The cycle mainly consumes acetate in the form of acetyl-CoA and water, which reduces NAD to NADH, releasing carbon dioxide, this cycle. And this NADH, which is generated by this citric acid cycle, they directly fed to or enter to the oxidative phosphorylation. The next topic after Krebs cycle we will discuss about oxidative phosphorylation. So this NADH generated directly enters to the oxidative phosphorylation or electron transport pathway. So the net result of these two closely linked pathways is the oxidation of nutrients to produce usable chemical energy in the form of ATP. In eukaryotes, the citric acid cycle occurs in the matrix of the mitochondria. But in prokaryotes, because of the lack of these organelles, they also lack mitochondria. The citric acid cycle reaction sequence are performed in the cytosol with the proton gradient for ATP production being across the cell surface. So the proton gradient for the ATP production is present within the cell surface, that is plasma membrane, rather than the inner membrane of the mitochondria. The overall yield of energy containing compounds from the TCA cycle is, so the overall energy yield is 3 NADH, 1 FADH2 and 1 GTP from one glucose molecule having 6 carbon atoms, 2 molecules of pyruvate having 3 carbon atoms each are formed during glycolysis. So in the last part we have discussed about glycolysis. So from one glucose molecule which has a 6 carbon atom, this glycolysis yield 2 molecules of pyruvate, each of them having 3 carbon atoms during the glycolysis. So 2 molecules of pyruvate are formed, and under aerobic condition If the bacteria involved in glycolysis have aerobic metabolism, the pyruvate molecule are converted to acetyl-CoA. If the current bacterium involved in the glycolysis are anaerobe, if it is an anaerobe, you think if it is anaerobe instead of aerobe, what happens? This pyruvate molecule directly enters the fermentation pathway. That's all. While one molecule of NAD plus it is reduced to NADH in the Krebs cycle, the acetyl group in the acetyl-CoA has 2 carbon atoms because carbon dioxide has been cleaved off and now enters the Krebs cycle. So the one molecule of NAD plus reduced to NADH and the acetyl group in the acetyl-CoA has 2 carbon atoms because the carbon dioxide cleave and thus enters, because actually the glucose after glycolysis we yield 2 molecules of pyruvate. So this pyruvate are unable to directly enter the Krebs cycle, so it may undergo a type of reaction and they are converted to what? Acetyl-CoA with the help of several enzymes. Okay, and this acetyl-CoA now able to combine with the oxaloacetate in the TCA cycle and continue the cycle. So this pyruvate is not able to directly enter. It should be converted to acetyl-CoA for entering the Krebs cycle. The Krebs cycle is a pre-step to electron transport chain. Now about discovery. Several of the components and the reactions of the citric acid cycle were established in the 1930s by the research of Albert Szent-Györgyi, who received the Nobel Prize in Physiology or Medicine in 1937, specifically for his discoveries pertaining to fumaric acid. It is a key component of TCA cycle. Next is about overview of citric acid cycle. The citric acid cycle, it is a key metabolic pathway that connects carbohydrate, fat and protein metabolism. The reactions of the cycle, and those are carried out by 8 enzymes that completely oxidize the acetate. It is a 2-carbon molecule in the form of acetyl-CoA into, and it is completely oxidized to 2 molecules each of carbon dioxide and water. Through the catabolism of sugars, fats and proteins, the 2-carbon organic product acetyl-CoA is produced. So it enters the citric acid cycle. So it is produced as a result of the catabolism of sugars, fats and proteins. The reaction of the cycle also converts 3 equivalents of NAD+. This I have already mentioned, nicotinamide adenine dinucleotide, into 3 equivalents of reduced NAD+. That is NADH, that is the reduced form. 3 equivalents of reduced equivalent forms. 1 equivalent of, so 3 molecules of NADH, 1 molecule of FAD, that is flavin adenine dinucleotide. Then into 1 equivalent of FADH2, that is 3 molecules of NADH, 1 molecule of FADH2 and 1 equivalent each of GDP, that is guanosine diphosphate and inorganic phosphate, into 1 equivalent of GTP. So GDP can be converted to GTP. The NADH and FADH2, those are generated by the citric acid cycle, are then used by the next pathway, Which is the next pathway? Oxidative phosphorylation pathway to generate energy-rich ATP. So these are utilized for generating ATP. One of the primary sources of acetyl-CoA is from the breakdown of sugars by glycolysis. So I had already told that during the glycolysis we yield pyruvate, and this pyruvate is not directly enter the Krebs cycle. So it may undergo a decarboxylation reaction with the help of the enzyme complex, that is pyruvate dehydrogenase complex, and this may generate acetyl-CoA according to the following reaction scheme. This is pyruvate, and in the presence of the carbon dioxide, in the presence of enzyme pyruvate decarboxylase, this can be converted to acetyl-CoA plus NADH plus CO2. NADH plus CO2. The product of this reaction, that is acetyl-CoA. It is the starting point for citric acid cycle. Acetyl-CoA may also be obtained from the oxidation of fatty acids also, not only from the splitting of the sugar, that is from the glycolysis, it can also be obtained from the oxidation of fatty acids. So this other reaction, look, this is the acetyl-CoA. Already pyruvate was converted to acetyl-CoA, then it gained entrance to the Krebs cycle, and this is the Krebs cycle, a cyclic form. So first of all, this acetyl-CoA combining with oxaloacetate, they are converted to citrate by the removal of a coenzyme A. Citrate now converted to isocitrate. Isocitrate by the removal of carbon dioxide converted to alpha ketoglutarate. Alpha ketoglutarate converted to succinyl-CoA, that is by the gaining CoA and removing carbon dioxide in the presence of, so the NAD plus can be converted to NADH. Here one NADH molecule is formed, and also here and also here. So that's why 3 NADH molecule. Then succinyl-CoA can be converted to succinate by the removal of this CoA. Here also one FADH2 molecule is generated. Then succinate to fumarate. Then fumarate by the adding the water can be converted to malate. Again the malate dehydrogenase enzyme which convert NAD plus to NADH. The dehydrogenase reaction which may result in the formation of NADH. Here, 3 NADH. And finally oxaloacetate is formed. And again this oxaloacetate repeat this reaction by combining with this acetyl-CoA again. Then the cycle is continued once more. Like that it takes place so many times, this cycle. This is citric acid cycle. Finally, component of carbon dioxide, that is 3 NADH, 1 FADH2, and also the ATP molecules, that is GTP. Look, during the conversion of succinyl-CoA to succinate, there was a synthesis of GTP occurring. GDP plus this phosphate, succinyl-CoA converting to succinate, this inorganic phosphate that combines with GDP to form GTP. So here the GTP synthesis occur. Like that. So the final molecule is carbon dioxide plus water plus this 3 NADH2, then FADH2. These are the intermediates in the oxidoreduction. First of all, we have discussed about the oxidoreduction pathways. In that we have mentioned about this NADH, FADH. Those are coenzymes, and these are important intermediates in the next cycle, that is electron transport chain. So these are utilized to the electron transport chain. Finally, the energy is formed in the form of ATP. So this is the overview.